So welcome to Butterfly Effect, an ecology of lepidopterists, which sounds maybe a little mysterious or metaphorical or poetic for, science and poetry, of course they go together. This talk began with a conversation. Actually, it began with a conversation between my father and Marc Epstein when my dad was declining this spring, and he called him up and he said, "Marc, I think it's going to be left to you to write my career obituary." And then Marc and I connected, and we started talking about all the intersections between, and Marc's going to expand on this, between what drew Marc to the U and their conversations, collaborations, aspirations. And it just became real interesting to me, the kind of the social life of science, the way that whole career trajectories and whole areas of study get affected by the friendships and the exposure to different ideas. So that was the beginning of this. And so, yeah, with that sort of, if you think about the stories you're going to hear today, they're not just about science, they're about people and relationships. And so this kind of social life of science. So in honor of my dad, in memory of his research, teaching, and love of nature, the Herbert M. Kulman Trust funded today's talk. But I want to also thank the important co-sponsors, the Bell Museum, you're going to hear from Holly, and the Minnesota Department of Entomology at the University of Minnesota, and the Harriet Alexander Nature Center, where you are, and couldn't happen without all of them. So just, you've probably all seen the flyer, but I'm just going to read the first paragraph to sort of set the stage. A small chance encounter. Well, you may know about the butterfly effect. He's going to talk about that. A small chance encounter can alter the life cycle of a lepidopterist, a person who studies or appreciates butterflies and moths. Like many of their winged subjects, lepidopterists often migrate from their childhood homes to their places of advanced education and onto their careers. Along the way, they form a web of connections with others that share their appreciation and curiosity about the natural world. These connections lead to friendships, new understanding, and sometimes new branches of study. So that's what we're going to talk about today. And I just want to introduce speakers. First, we're going to have Holly. Holly Menninger is the executive director of the Bell Museum, and she leads the Minnesota State Natural History Museum, which is the Bell Museum, and Planetarium. In this role, she's responsible for the implementation of the Bell's mission to ignite and sustain curiosity for nature and the universe through exhibits and programs that engage diverse audiences, scientific research, and curated collections that investigate biodiversity locally and globally, and statewide outreach that meets communities where they live, work, and play. A science communicator by passion and practice, she has long worked at the intersection of science and society in policy, natural resource management, and public engagement in science. And I'll just introduce myself later when I say a short piece. Actually, I'll introduce you, Marc, before you speak. That might be more. I'll let you take the mic there.
Right. I don't want to trip over stuff. Yeah. Hi, everybody. Are we alive? It's so nice to see some familiar faces in the audience, as well as some new faces. Thank you, Janae, and to the whole Kulman family for inviting me to this special event in honor of forest entomologist Herb Kulman. I'm sorry that my path has never crossed with Herb. I'm a relatively recent transplant to Minnesota. I came here about 7 years ago. But from what I've read and what I've heard from Janae, what she's shared, it sounds like we might have had a few important things in common. One, I think we both have and had a deep appreciation and curiosity for the natural world. And I also think we probably shared a passion and drive for sharing that with others. In Herb's case, sharing that with his children and grandchildren, as I think we'll hear, his friends, fellow garden club members, and then, of course, the 30 graduate students that he mentored over the course of his distinguished career. Herb's specialty, for some of you who may not know, was Lepidoptera. I'm not sure you know what that word means, but that's the order of insects that includes butterflies, moths, skippers, which I think are underappreciated member of the Lepidoptera. I'm seeing the entomologists shake their head in the room. This order of insects, I think, has really captivated humans for thousands of years, doing so with their beauty, their grace, and then, of course, their really interesting life cycle of complete metamorphosis. I had to put a caterpillar up there, too, because, we're talking a lot about butterflies. I'm not sure how many caterpillars. Oh, we'll see caterpillars in your talk today. I think for many, butterflies are really a gateway insect to the study of entomology. That sounds a little funny, but it's true. And that's my kind of the theme of my talk today, about butterflies as a gateway. They're accessible, they're colorful. When you're thinking about public engagement in science, they don't bite or sting, which is usually somebody's first concern when we're talking about an insect. And they've really captured the imagination, starting with really young age, including my son. This is my son actually here at Harriet Alexander Nature Center. We come here all the time. And this is my colleague's daughter Sage, who reared black swallowtails courtesy of one of our Bell Museum volunteers. Scott Magnuson always supplies us with caterpillars and chrysalis to raise. I think I've had the great pleasure as the director of the museum, and then previously as the director of public engagement, Director of Public Engagement and Science Learning at the Bell, to really see this play out, this curiosity and fascination with butterflies play out daily at the museum. So particularly in our Touch and See lab, where we often have live specimens, or we'll have chrysalises on display, and then certainly have some collections for our visitors to interact with. This is one of our student interpretive guides. It was very much on display last night that we host our parties every month, but last night was our special mothing night. We do this once a summer as part of National Moth Week, and we set up black lights, and we invite our visitors to come in. And even though it was a chilly evening and there were only a handful of noctuid moths coming to the light, people were fascinated. They were crowding around the black lights, leaning in, asking us questions, trying to follow their curiosity and learn more. I think for me, in many ways, this was me many years ago. Also, this is like an original Nirvana t-shirt. I'm just going to point that out for some of us in the room. That's not like the vintage ones you can buy at Target today. Butterflies were really my gateway insect, the taxa that really steered me to the study of entomology and to a career focused on public engagement in science and a real passion for natural history. So I spent the summer after my sophomore year of college doing a research project. I went to Denison University in Central Ohio, and I worked with my advisor and a couple of other undergraduate students to learn about and survey the butterflies of Central Ohio. This was part of a long-term biomonitoring project. At the start, I did not know much about butterflies. I knew monarchs. That was like the one thing I could identify. But I was very lucky to have an amazing advisor named Tom Schultz. Most of the really beautiful pictures that you see here on my presentation are pictures that he took. He's a fantastic insect photographer. And with Tom's guidance, with many hours spent with this book, Butterflies Through Binoculars, and then doing many what are called pollard walks, some of you know what those are, through the trails of the Denison Bioreserve, I got to know our local species and learn how to identify them on wing. It's where I first learned the word gestalt. Entomologists are where you get to understand sort of the way that a particular species flies and interacts with the landscape. And it was such a joy to become acquainted with these beautiful creatures that I don't think I really paid attention to before. Things like the red-spotted purple, the banded hairstreak. I love this one, the great spangled fritillary pipevine swallowtail, among others. And that summer was incredibly transformative for me. It really opened my eyes to this biodiversity in a place that I thought I knew, but I didn't really. It piqued my curiosity for ecology. It motivated me to ask questions about the patterns I was observing in nature. So whether those were differences in populations I observed from different sites or amongst species themselves. And it certainly then made me a lifelong enthusiast for butterflies. In fact, I drive my husband crazy every time we go on a hike because I'm always calling out the names of the species that I see, and I feel compelled to say that out loud, and he finds that maybe charming. I don't think so, but that's okay. That summer really, I think, solidified my identity as an entomologist and ultimately inspired me to pursue a PhD in ecology and entomology. And so while my research interests, I know, not butterflies, my research interests shifted largely to aquatic insects. Hi, Dean. It's nice to see you. So shifted to aquatic insects, particularly dragonflies and damselflies, and then on to 17-year cicadas. I really do credit butterflies for first piquing my interest and deep appreciation for insects and natural history. An interesting kind of coda to my story, and that's sort of my first dive into entomological research. Although I didn't know it at the time, the work that I did surveying the butterflies in Central Ohio really led me to a movement and an approach known as citizen science, sometimes now called community science, which is where professionals and the public partner together to create new knowledge. It's an area where I spent a lot of time later on in my career, but sort of early on, I don't think I was wholly aware of that. And the really cool thing is that our data that we collected on a few routes in Central Ohio really joined those collected by other professional scientists, amateur scientists, trained volunteers who were all part of this large-scale monitoring project that was organized by the Ohio Lepidopterists Society. The extra cool thing is about 5 years ago, former colleagues of mine at NC State University, where I was before I came to Minnesota, published a really important paper summarizing those decades of monitoring data that were collected by people like me and other folks across Ohio. In fact, these are my cites. There were lots more people that my advisor and other students continued it on afterwards. And so, while the title of the paper suggests, the results were a little bit grim because it was about population decline in butterflies, I was really proud that those efforts from that summer contributed to something bigger than me and that contributed to this larger scientific enterprise. I think, thinking about citizen science and where citizen science intersects with Lepidoptera, we live in a really exciting time where there's opportunities for you to participate in the study of butterflies and moths and skippers to contribute to something bigger there. And this is just a few projects that exist out there. I was trying to list, there's too many to list. What I thought was fascinating is on the platform iNaturalist, I usually just use the app, but I went to the website and I typed in Lepidoptera, and there were over 24 pages, and each page had 30 listings of projects for studying butterflies and moths and skippers. And so I thought that was really exciting, and now is the time. So I'm hoping that maybe one of these projects, I'm looking at sort of the younger people in the room, might be your gateway for deepening your appreciation and expanding our collective understanding of Lepidoptera. So thank you. Okay, trying to get out. Mark, I'm gonna set you up. All right, here we go. Oh, you're next, that's right. Sorry, Jenny. Do you want me to introduce you officially, or are you gonna introduce yourself? Hey. We will scratch each other's back. All right. Well, I am pleased to introduce Janae Coleman-Brigham, Herb's daughter, and a senior research fellow at the university, who we can thank for bringing us all together today. Janae will provide a brief child's eye view of growing up with a forest entomologist and lover of butterflies. Do you have a picture pulled up that you want to see? Or?
No.
Okay.
here, we'll just, okay. So I thought I would just share little glimpses, little windows into, we're talking about the social life of science. That social life extends to the home. So I guess as an origin story, of course, not of course, but my dad met my mother at the University of Minnesota when he was getting his PhD. So that's how I began. These are just little vignettes. The way that just his orientation to nature and butterflies in particular, I have this particular lens on his butterfly dimension of his life. When he would tell us stories as kids, if there was a butterfly in there, his hands would start to fly around. Butterflies, and I did that with my kids too. And sometimes the butterflies I don't remember if he did this, but they do the little thing. Yeah. So this kind of embodiment. On our fireplace, all around, right on the brick, were mounted many different framed page spreads from a butterfly field guide. So they were there. We went butterfly collecting. I was trying to find this isn't the original one. We have the aluminum, extension net. And I learned, we didn't go out hunting for deer. We went out hunting for butterflies and pinned them. And I had the little cigar box collection, like so many kids do. This one's a bit of a stretch, but I look for patterns. So he was a very good swimmer. He did the butterfly stroke. Okay, I'm just going to say he didn't name that, but he had big, powerful arms. Michael, my brother Michael, who's here, remembers that my mom was a smoker, that he would save the plastic sleeves off the cigarette cartons so he could use them as collection envelopes and fold, when he was out for insects or plants, and have them in his pocket. He was always collecting things. While not necessarily about butterflies, just this orientation to nature, we had the celebration of life here last night, and I was very much at home with all the taxidermy, because in our living room, there was a moose, 2 ram's heads, the front half of a goat on a promontory, stuffed owls, stuffed frogs. We had, it was like a natural history museum in our living room, which my mom, who was an interior designer but liked to be interesting, let's go with it. One thing that I'm really curious about, I hope with conversations with his grad students I can learn more about, is in our garden, he had window screens tilted up around plants that we informally called the butterfly cages. And I never really thought about it much till I was reflecting on the role of butterflies in his life. And I think I must have thought in the back of my mind that it enclosed butterflies, but I believe that it was protecting caterpillars from predators so that he could study. And I am curious if this was part of anything that went into the butterfly production management. I don't know. Maybe we can learn that later. But one of the things that I think, butterflies, but also just nature in general, that is one of my greatest gifts from my father, is just, going on hikes. And some people hike and they're trucking along, right? But we'd stop and we'd look at details. We'd go slow so we could see things. And the whole world was open for interpretation. And not just interpretation and observation, but appreciation. And so I wrote this for the celebration of life last night, but it's just as applicable here in this appreciation. So I'm going to conclude with this poem. Butterflies in the sky. Isn't that wonderful, you asked your children, as 2 cabbage whites danced in the air? Isn't that wonderful, waiting for the red admiral to land and flap its wings, flap its red stripes for our admiration? Isn't that wonderful, the swallowtail with its exotic wing extension and fancy yellow dress? And isn't that wonderful, the double eye spots on the modest brown wood nymph, the lemony clouded sulfur, the luminous monarch with its tales to tell from far away? Each encounter was a call and response. Yes, oh yes, that is wonderful, we agreed, praising the fleeting colors of butterflies in the sky. So those are just a little window into child of lepidopterist, or really entomologist. With that, I'm going to introduce Mark. Are you going to talk about camping in the yard? No. Okay. Well, I'm going to introduce him then. I met Mark, I don't remember him vividly, when I was a kid, and Mark came to the U, and there was always grad students around our house growing up. And some of them slept over for a couple weeks or a month. Yeah, camping until the apartment was ready, so we had people all around. So I guess I've known Mark a long time. So Dr. Marc Epstein is a senior insect systematist, thank you, at the California Department of Food and Agriculture for Lepidoptera, moths and butterflies. Dr. Epstein has worked for the last 20 years at the Plant Pest Diagnostics Center in South Sacramento. The modern lab includes dozens of scientists and technicians for entomology, plant pathology, seeds, nematology, and botany, all whose mission is critical for protecting California agriculture from plant pests. He is also a research associate at the Smithsonian Institute's National Museum of Natural History, where he was a postdoc and on staff for 15 years. While identifying Lepidoptera of potential threat to agriculture, he researches their evolution and classification with an emphasis on the moth family Lemicodidae. And, okay, sorry. And the history of entomologists who were his predecessors. You're the featured speaker, so I'm going to do the extended bio here, so in case people haven't read the brochure. He authored Moths, Myths, and Mosquitoes, The Eccentric Life of Harrison G. Dyer, Jr., published in 2016 by Oxford University Press, which you can buy and have signed and enjoy after the talk. And he received a PhD and an MS from the Department of Entomology at the University of Minnesota, 1988 and 1982, and a BS in entomology at Colorado State while on a music scholarship in 1977. Outside of work, Mark continues to perform and teach clarinet around Sacramento, performing klezmer, classical, and Latin jazz. He is active in iNaturalist.com to teach and encourage raising caterpillars around the world, particularly of his limicodid moths, for the purpose of determining their identities. He continues his butterfly survey work in his native Colorado at Roxborough State Park, which he began in 1979. Dr. Epstein has been a guest on NPR's Fresh Air with Terry Gross in 2004 about his work on the book Night Visions, The Secret Design of Moths. And there's links to more about Mark on your flyer. So with that, thank you, Mark.
Thanks so much, Jonay, for all of this. It's really been something. I've been fortunate to stay in touch with Herb all these years, and I was a little frightened when I received that note that he wasn't long for this world. So I called him right away, and he was still very much with us, but subsequently to his passing, getting to know Jonay has been such a pleasure, and so much of Herb's energy I just feel in her, that creative, can-do kind of can-do part of his personality. Wow, I skipped that many pages. So today I'm gonna, on the tails of the monarch, even though monarchs don't have tails. I'm going to talk about how I came to be here in Minnesota, and about Herb and about his butterfly production management. And so Oh, I'm going to get used to this. Okay, yeah, so the butterfly effect, the idea that the beat of a butterfly wing can travel across the planet and have some kind of influence on things. And I love the way Jonay thought about it in terms of how lovers of Lepidoptera interact and around this thing, because it is kind of almost magical, really, how we connect through all of this. So here's, you can see, is this a butterfly effect? No. I knew John would know. So this is actually what's called a swallowtail moth. And this was at a UV light, or actually a mercury vapor light in Vietnam just about a month ago. And like our swallowtail butterflies, we often had some that were missing the tails. So they had been attacked by something, possibly birds, possibly lizards, something. So I want to move on. So this is a species, Lissa zampa, and I was quite surprised that it didn't, given how common it was on our trip, very little about it on iNaturalist. So I posted pictures of it so you can look it up under there. So the butterfly effect, could be a way we could say that Herb actually brought me here to Minnesota. And then I'm going to talk about Limacodidae, the slug caterpillar moths, and they can be pronounced limacodid, or limacodid. I tend to say limacodid, but it's just like you could say Epstein or Epstein with my name. So anyway, quite interestingly, this was not what I had planned, but my study of limacodids for my dissertation led to me becoming a biography of a very notable eccentric entomologist, Harrison Gray Dyer. And I can't quite imagine how people would have solved some of these mysteries without studying Limacodidae, and I'll get to that later. So I'll also give some present examples of new discoveries about slug caterpillar moths, many that have resulted from collaborations via iNaturalist. So my last visit to Minnesota was in 2017, and some of you were there, and I gave a lecture for the Hodson Award that I received. And I mentioned how he brought me here, but I didn't really talk about how kind of the circumstances in any great detail that butterflies actually played a part of it. So you're going to get that. And then I'm going to end my talk with a bit of history about Dyer and, about the history of, I'm going to talk about Dyer too, but first let me get into a little bit of Department of Entomology history. So this was in 1982, and I actually saw this. I was walking from the student center, and up the hill was Alec Hodson taking little notes about the phenology, which he did for 40 or 50 years. So every little flowering event on campus, he would record, and even had surrogate plants in preparation in case that one would bite the dust. So there he was. I think he was smoking a cigarette, but I'm not positive. That might have been in my imagination. He did have an ashtray under his portrait in Hodson Hall, which I always thought was funny. But anyway, here I see Hodson. This I know. And then down at the bottom of the hill were, the tall guy was Richard Jones, and the middle was Milt Weller, and then next to him was Ira Adelman. So there was the three of them in their suits, some of them three-piece. I think Weller had a three-piece suit. So what that marked was actually the dissolution of the Department of Entomology, Fisheries, and Wildlife. It became Fisheries and Wildlife as a department to be headed by Ira Adelman, my music buddy that I played music with in the basement of Hodson. And Richard Jones took the entomology reign. And that was quite a freeing experience because prior to that, our resources were always kind of like with students, like, okay, this year wildlife will get to go to a meeting, next year fisheries will go, and then entomology. So it was this sort of frustrating, and it wasn't exactly a natural department. But I do kind of wonder too about the butterfly production management that I'll be talking about, whether being with wildlife and fisheries, whether that had a little bit of influence on her thinking in this manner. So I talked in that lecture about my career and, oh, I'm going backwards. Okay, so anyway, speaking of iNaturalists, so back in 2019, there was 21 million observations of, I like the term community scientist, by the way. Citizen scientist has always kind of bothered me, so thank you for that. So there were 21 million observations. And then, lo and behold, in 2024, it's 10 times the number. And this is of all plants, all insects, all animals, everything. It's just been an explosion. And actually donate to it if you can. I'm a little concerned about they've become an independent organization again, and whether or not they can survive. That would be terrible if they don't. Because the amount of scientific information, even though we can access it and download it, not all of it. And I've been working with them. Their developers are in the Bay Area, so I've worked directly with them. It's been a little bit difficult. So now about the Limacodidae, they're really made for iNaturalists because not only are the caterpillars interesting, but the moths are too. People just tend to really photograph them. A lot of people say, oh, the moths are not anything, but they have a lot of weird postures and they can be pretty colorful and have neat patterns. It's just that the caterpillars are so unusual. They're sort of like terrestrial nudibranchs, the sea slugs. More importantly, though, behind each one of these images is a person that has photographed it, and in the case of the caterpillars, potentially could raise it. A lot of times these are done in real time, so I write to these people, and maybe it's 1 out of 30, but I get certain people that have already kind of caught the Limacodid thing, and and sometimes it's a few years, but like I'll give you an example. There was a moth that I know from Costa Rica that is a mistletoe feeder in the larval stage. Very unusual thing that Jansen discovered. I guess mistletoe got blown off of a tree, and a keeper from the Natural History Museum London found this really weird larva. And so anyway, this photographer in a nature reserve in Brazil sends me a picture of the adult, and I said, look at your mistletoe, maybe you'll find the larva. And sure enough, within 6 months, I get this amazing larva, and it's only the second time we know. So we know this larva from Costa Rica and Brazil, but that moth also occurs in southern Arizona. So this is kind of the power of iNaturalists. In the old days, sometimes I get a letter, can you identify this photo? And, the turnaround of these interactions was so slow. I mean, it was special, but it was slow. So as a result, I'm starting to get students. I don't have an academic position, but I'm getting students through iNaturalists, and I'm on their committees. Luke, on the right, he's getting a PhD at George Washington University. And then Guilherme Fischer is at the University of Brasilia, and both just visited me. I went to Luke to Vietnam. I've been on 5 trips with him now. And what was really interesting about Guilherme is that during this visit, he said something quite poignant. He said, when we talked on Zoom the first time, I was already really interested in Limacodids, but you asked me, am I interested in music? And he said, that sold me on it's like I hit a, I hit a chord, but sorry. I'm sorry, Joey. My son's here. He has to put up with this. But he really remembered that, and this is kind of what we've been talking about with Herb. I didn't always see that part of Herb in our, it was later, during later part of my graduate career when I was at his apartment and he was playing, believe it or not, Herb Alpert, and cooking rutabagas and all kinds of things and telling me that my car needed washing, that he could see down below. But anyway, Herb really had a special side that we didn't always see, and I fortunately got to. So Guillermi is going to finish up and go to graduate school. So, and then there's two others. There's one at Oxford right now that's getting her PhD, and then a fourth already has her doctorate, but she's in Taiwan. So I sort of have these four musketeers that are learning from me on iNaturalist and in other ways. These are all pictures of larvae that we got from eggs in French Guiana. Luke we actually have permits at GW to rear these caterpillars. The only problem is the permits tend to lag way behind when we actually get the stuff, but so it goes. And this was, there is kind of a decline in moths and butterflies, it seems. Though this trip, it was pretty rich, and it turns out French Guiana actually markets the idea that at its various, I don't even call them reserve, they're like private reserves. They provide mercury vapor lights, and lots of people, especially from France, come to get specimens, and the permits are very lax there, just as long as you bring home like 1,000 specimens or less, and that's about all that's required. I tend to require a microscope when I go because I'm preparing Microlepidoptera, because usually that's what my source of funding is for, like tortricid moths and small things. Plus, I don't know all these groups, so it's nice to be able to look at them under the microscope. So what do you think we're seeing here? I got this picture with a text from Brazil a few weeks ago. What do you think? What kind of insect are these? Yeah, there's more than one. What do you think they are?
Ticks.
I mean, when I first saw this, even though I should have known better, I thought I might be seeing a cicada on the left, but then the one on the right, I mean, I recognized it as a moth, but I didn't quite get it it took me about 10 seconds. So this is the fellow that sent it to me, and he's actually number 5 student, Saulo from Brazil also, and he's probably a year behind Guillermi. Just amazing. So what we found is that this is a new species of lima-coated moth. The one on the left is a male clearwing, and the one on the right is a female. Well, I knew the clearwing already because at the Smithsonian, there were specimens of it that were, and nothing about the caterpillar, but the moth had a tag on it that said it would fed on eucalyptus, and that's all we knew about it. But then Guillermi figured out, and Saulo figured out the female from rearing the caterpillars. They would never match those two up. And that has occurred in all lineages of lima-coatids practically. You have a clearwing male that goes with a female. The hag moth here in Minnesota does something very similar. And with the hag moth, you actually get more females at lights than males, which is the reverse of what is typical. It's because they are day flyers. The males are. So this is the other part of the story. Where's this little pointer? So, can you see that up there? I can't even see. Yeah. Okay, so anyway, the female emerged from this cocoon. It crawled or walked all the way over here, sent out its pheromone, and this was during the day, and it attracted the male, and they mated. So this is like an observation that Saulo made. So not only do we have the male and the female, but we have this biological stuff. And we've always thought that clear wings were more for the day for reasons I said. And here's what the larva looks like. So you're the first people to really see this, besides a few of us. So that's the egg mass on the left, and then a early, probably second instar. And as we'll see, the first instar, the ones that hatch, actually fast and then molt the next day. And then that's like a third or fourth on the right. So, yeah, go ahead.
Did you say that the host plant is eucalyptus?
No, that was a record for it, but actually the natural one is something else. And
Eucalyptus is not native.
no, of course not. Yeah, which is actually interesting because there have been other records from South America on eucalyptus as well, but clearly not. They've switched to it, and of course in Australia it's a favorite. But yeah, it's I guess it's pretty suitable. So to put this a little bit in context, I've been working on the tree of life, the phylogeny of Limacodids, using both molecular and morphological. So essentially this new moth fits right here in this part of the tree. But what's really interesting is just a few days prior to getting these from Salo, we were in Vietnam and we got a sister group. So there's a connection, which I already knew from the genes and from the morphology, between caterpillars in Asia and in the New World. And the New World ones are all tropical. So it's really an old connection. So now a bit about how I came to be a naturalist. We've heard a bit about that. Quite interesting to hear other people's stories. So I was fortunate to come from a family that encouraged my interests, and we went up the mountains a lot, so that didn't hurt. But my Jewish ancestors just happened to come to Colorado because of tuberculosis, actually, at least on one side of my family, and there was a pioneer family on the other side. But it turns out Colorado is actually one of the most speciose states for butterflies. It's just an amazing place, more than California. Probably not as many as Texas, but in terms of native species, it probably is the top. So this is where it all began, at my home at age 4 with my brother, Alan, who has the net. He captured a painted lady, which tend to migrate to Colorado every 4 to 6 years. So I can actually backtrack to the being 4 years old, even though I know it is for other reasons. But anyway, I was just totally mesmerized by seeing one of these butterflies in a glass container where after we captured it. So this is kind of simulates it. So that light shining through the wings, I just could not believe it. And from that moment on, I imprinted on butterflies, no question about it. These are from my earliest collection, a red admiral, an Edwards fritillary, and a common sulfur. And then when we go to the mountains, first I used to hate it because of the drive, but when I realized that we didn't get the same butterflies there, that changed everything. That one on the bottom right is the Colorado hair streak. And above that's the short-tailed swallowtail. So at age 10, this became my Bible, and it was written by F. Martin Brown, F., who was a postman, and Roger, who was a priest, a Jesuit. I got to know Brownie when I was 10, but then as I got older, I was able to get him to come talk to Colorado State University. He was very interested in history of entomology, which is probably how I got interested in it. He had this project called the Four Station Project, which he continued through his 80s and early 90s, where he did biodiversity inventories of 4 different places. One of them was the Great Sand Dunes. My first job was paid to collect butterflies, actually, which is kind of amazing, at the place that I hung out anyway most of my life. Now it's called the Denver Museum of Nature and Science. And when I was 21, the assistant director asked me to plan a phenology exhibit on the butterflies of the famous Red Rocks Park, and we found nearly 120 species occurred there. Sadly, the exhibit never happened. I guess some donor didn't like the idea of this butterfly exhibit in the middle of a hall with dioramas or whatever. So, but the good thing was that I got a scientific paper out of it. So once I hit Colorado State, I was fortunate to have Howard Evans as my advisor, who had written this book, Life on a Little Known Planet, Wasp Farm. He was actually a tenured Harvard professor that decided to leave Harvard because he wanted to study his wasps. So he came to Colorado State, and I just happened on him as my advisor. So he loved the clarinet, which really helped me be able to get this music scholarship, which was offered to me. He got me to not have to take certain things like sociology, which maybe I could have used, but I took music theory and had a senior recital and had the whole music curriculum. The other thing about Howard that was interesting is that and we'll get to this in a minute, from Life on the Little Known Planet, which Herb quoted in his Butterfly Production Management, he talks about, how sad he was at losing the butterflies that he grew up with, from habitat destruction. But what was funny is he used to tease me about butterflies. He'd say, because he was an ethologist, he studied the behavior and the evolution of really interesting sand wasps. So he would go, oh, butterflies, all they do is fly around and look dumb. But then I could say, well, Howard, the Xerces Society has Howard Evans, quote, butterflies matter to me. So I got him back. So graduate school, that was kind of an interesting thing, because even though I had 7 summers of museum experience, I had a bit of a problem because my GREs weren't so great. And my entomology was great, but a few bad chemistry grades, whatever. But my mentor, Bob Pyle, who founded the Xerces Society, Bob is also from Colorado, he mentioned Herb as a potential advisor. And he was particularly enthusiastic about the Butterfly Production Management paper from 1977. So I kind of like the name BPM, like IPM. But anyway, Butterfly Production Management, the idea is to alter habitats to enhance larval and adult butterfly food plants, hibernation sites, volatinism, multiple generations, or be able to consider how many generations, fecundity, how many eggs they lay, adult longevity, and also just to increase the accessibility to people so they can see these amazing creatures. But part of what was interesting was the idea to introduce species that are scarce or absent early in the season because winter mortality may have taken them, or they might be slow to immigrate into an area. He also boldly suggested exotic butterflies and larval host plants. So in the Butterfly Production Management paper, Herb cited works of Evans, Brownie, who I mentioned earlier, F. Martin Brown, and Pyle. And in his, he quoted Evans, or a quote from Herb is, in his interlude in the Elysian Meadows, Evans lamented the apparent decline in butterflies around the world. he despaired at finding butterflyless house-filled suburbs in meadows where he chased butterflies as a child. And of course, I had that same experience in Denver, places, both Bob Pyle and I had our own spots for the painted crescent that were destroyed by shopping malls or whatever, groceries. And from Brown, he talked about, Herb mentioned about the pipevine swallowtail, that as being a possible exotic that occasionally gets to Minnesota, occasionally to Colorado, but it's kind of an example of something that's not going to cause any problems because it's on a very narrow host plant and so you could introduce it and not worry about it. So this was a statement from Herb. I will not promise that we can return Evans Meadows, but I will suggest that with the use of applied research procedures, we can develop management methods to provide productive havens for butterflies in suburbs, other disturbed areas, and possibly in some natural areas. And Herb sent this letter out with the article, and I guess I got it too when I became a student. And it was kind of an interesting letter because, he was trying to extend the reach of the paper because it was published in the Minnesota Ag Experiment Station Bulletin, which is not well known. But also, at the end of the letter, he wrote something curious, and I think it really not only reflects kind of his own reservations, but a concern for his students doing work on butterflies. So what he wrote was, for the future, I plan to conduct research on several of the items mentioned in the paper. However, it will be only a part of my research activity since I have neither the inclination nor the mandate to significantly reduce my research in biological control of forest defoliators, which has been my entomological raison d'etre for the past 22 years. So kind of interesting, and Herb did not pursue this, so I did want to mention. But by the same token, this paper did attract me to him to begin with. So Herb also had become interested in carabid beetles, and he saw that as a potential for biological indicators. Also, they were predators, so they could have some beneficial aspects. He also had already done some work with Robert Dana in some nature conservancy natural areas. Thus, he offered me an assistantship with the beetles, but not the butterflies. So in spite of having work experience at the museum and excellent coursework, I guess the music was kind of a deterrent. People didn't think I would be serious entomologist. So I didn't have an easy time getting into a program that was funded. I got into several programs, but not with money. So Herb mentioned to me about Robert Dana, a passionate lepidopterist at the U of M that he knew, and that I thought, well, with some money for the beetles, a couple people interested in butterflies, that would work out. But there was one other little thing that Herb mentioned, and that was that starting in the fall, Paul Swedenborg, a fellow Colorado State University graduate that I knew, was also going to be starting up in graduate school. Paul and I had been on a Colorado State University trip to Baja in January '76, and there were 3 vans. 2 of the blue ones were entomology vans. The green one was botany, and Paul was a botanist at that time. One of those blue vans tended to be off on some mountain somewhere lost. Ours was kind of by a future, kind of like a Ken Kesey character, Carolus Bagdonis, who became known for Bagdonis's Flying Circus. But anyway, those 3 vans went to Baja, and then at the end of the trip, the 2 of us decided to hitchhike from Tucson to Los Angeles together on the way back. And unfortunately, we had lost track of each other. Paul got married, and I don't know, we just lost track. But then until I heard from Herb, and I thought, wow, that's really cool. So my master's was on carabid habitat and work and was a pitfall trap study at Arden Hills Arsenal. And I had up to 150 traps each week that I had to pull material from May to September, thousands of beetles to pin and label and identify. And these were all tallied using computer punch cards. And then fortunately, Bob Barteld, who was the senior member in our lab, he wrote Fortran programs to tabulate all this. This was before Excel, of course. And although I came to love carabid beetles and their habitat affinities, and they have amazing, when you look at them under the microscope, they just have such amazing diverse surface textures and all sorts of things that you would not imagine. I would return to Lepidoptera and never turn back. So I thought until 2004, not after long, I started in California at CDFA, I had a chance meeting with Kamal Gandhi at our museum. And when our coleopterist mentioned my name to her, she started jumping up and down and screeching because she knew my work on carabid. She was doing carabid work. She was the next person to do carabid work in Minnesota after me. So I told her what I've told people over the years. I really loved working on them, but the only problem is for my study to really mean anything, somebody'd have to go back and do it again. So Kamal, who then was in Davis, finishing up her degree in Davis, said, well, she said, oh, we'll do it. I'll get funding for it. It'll all work out. And I go, right. I couldn't believe her. And she did. She went to Robert and Dana and got funding through the DNR, and we did it. We did a 25-year comparison of the beetle fauna. Okay, so now some closing thoughts on Kulman's butterfly production management. And this came from Bob Pyle just a couple days ago. As a forester and biologist, conservationist and butterfly ambassador, I love the democratic nature of Herb's idea, which was truly iconoclastic in conservation thinking of the time. Let's use the biggest ecological canons we've got, not only to save the rare taxa, but to build up the common ones, to cultivate a clientele for appreciation and conservation of our rich natural butterfly heritage as a whole. This was new, and Herb came up with a great syncretic name for it, butterfly production management. While some rebelled at the seemingly mechanistic savor and others might today kvetch about details like his thoughts on exotic introductions, now an anachronism, his essential concept was as powerful as Dr. Jeremy Thomas's in England when he saved the endangered black hairstreak and reintroduced the extinct large blue through applied research. His idea was to build up butterflies, both to delight and enlighten people. It was, in a word, genius. Robert Michael Pyle, founder of the Xerces Society for Invertebrate Conservation and honorary life fellow, Royal and American Entomological Societies. So now we're going to return to Lepidoptera. So leaving carabids was actually not going to butterflies, but going to moths. And I didn't really get that excited about moths until I went to New Caledonia with Mike Pogue. That person sticking his tongue out at me is Gordon McPherson, who was the botanist on the trip and quite instrumental in getting us around. In New Caledonia, there was a caterpillar that actually found me as I was walking through the bush. It stuck to my clothing. I thought it was a lima-coated, but later I found out it was a related group called Lacturidae. Lima-coated or not, though, the seed was planted, and I had learned about saddleback caterpillars as Herb's TA in forest entomology, which I taught for 2 quarters. My dissertation led to my Smithsonian postdoc on Limacodidae and related moth families. The phylogeny and study of the evolution of the underbelly and associated morphology of the spinneret, the silk-making apparatus, and behavior was a big part of the study. Oops, there we go. So they're able to grip with these sucker discs and a very elastic cuticle underneath, very thin elastic. Can you see those little suckers? This is actually, all these examples I'm about to show occur in Minnesota. This is this type. This one from Colombia, you can see a similar type of locomotion. So the idea is underneath, and with their silk, their spinneret, they're pretty similar. They do a similar thing. It's the part on the back that's so different. So how they defend themselves, how they look, it's quite a diversity of forms. So here we can see a spinneret of a what's called a jewel moth, or now it's going to be a subfamily of Limacodidae. I've done a evil taxonomic deed. So these lay down, it's a the silk doesn't ever truly dry because they crawl right over it. And what part of the beauty of this whole system is because they lay down this sticky silk, when they have stuff like particulates getting onto their bodies, they crawl over their double stick tape of silk and it pulls it off. So that's how they can remain this way, how they don't get completely, covered with dirt. The other thing about the spinneret that's amazing is you can see there's brush parts, and that's actually the silk gland that produces the silk. Okay, so here's a classic limacodid. This was just taken a few weeks ago. This is from Vietnam. I call these fishtail spinnerets because they're shaped like a fishtail. And through electron microscopy, you can see and even these are not all the same, and sometimes they change as they grow. Sometimes they remain the same. All aspects of these caterpillars are quite something in terms of what we call ontogeny, which I'll get into a little bit. Okay, so putting it simply, limacodids all stages are cool. I mean, their development of embryos, the cats, the cocoon construction, the adult postures. This is one again from Vietnam that just hatched a few weeks ago. It's now probably almost full grown. So what we're seeing here is what I dubbed retractile feeding. So limacodids and their relatives, what they do is their mouth parts are hidden underneath their thorax. So kind of like a turtle, their head is tucked inside, but it's chewing on the edges. So part of the whole idea is to not be detected by predators. And conceivably, it also could muffle the sound as well, because caterpillars can be pretty noisy, especially large ones. These aren't so big. We're talking about caterpillars that don't get any bigger than this. So you could call retractile feeding a method of crypsis or defense, but then got all kinds of defenses with their spines. So this is the type where the spines spring out, kind of like a sea urchin. And they're venomous. So that's another aspect of this project that my students are going to be working on, is looking at the genomics of the venom. And there's a lab in Australia. They already have published several papers in proceedings of the National Academy of Sciences and Nature. And so another aspect of the defense are these things. Do you know what a caltrop is, or a caltrope? No. Okay, it's a medieval, those those spiky medieval weapons that they would, yeah, kind of, and they'd throw them, really a horrible thing. But these caterpillars have these patches of these deciduous spines that, and actually I'm pretty sure they're just like pollen. I sneeze when I'm around these, so I'm pretty certain, because they're so tiny and they come off so easily. So the whole idea is a would-be predator gets a mouthful of these caltrops, and not very pleasant. So I've also been studying the development of the caltrops. When do they appear? But also the caltrops only occur in certain lineages and then only occur like by middle stages of the caterpillars. So here's another type of defense, and these are in the gelatines, the smooth type. So these type of droplets, these are being analyzed finally by that group in Australia. So we're knowing something about at least the proteins of these little droplets. So here's our local monkey slug that probably occurs right here at this nature reserve. So they do the escape, kind of like, lizards do when you tug on a lizard's tail. This one's going to go forward and backwards to show you. Yeah.
No animal was harmed in the making of the
I plead the fifth. Okay, so this is a case where the wasp is harmed. So this is from Shannon Murphy. So yeah, they don't have much luck with this saddleback. And these, of course, occur here too in Minnesota, saddlebacks, like this one.
lost mark with page.
Yeah. So it's going to give up and try the cutworm in a second. Of course, I have others of the caterpillar that have been attacked by parasitic wasps. They don't do so well. Okay, get the cutworm. So even the eggs of these things are cool. And so the ones at the bottom are more the typical, most of them are like this type where it's the thinnest eggshell known in Lepidoptera. But more work needs to be done, like with the histology of this. But you can see the entire embryological development, which is amazing. I mean, there are other microleps too that you can do this with, but these in particular are so visible, as are the underbellies of the caterpillars as well. You can see the kidneys, the Malpighian tubules, the tracheals, all the different working parts. It's like they're the visible caterpillar, kind of like the visible, what was it called? The visible man, the visible woman. Do they still have that? I don't even, it's probably on video now, not on. So this is a few days old embryo of the genus Samira, which actually I was with Roger Blahnik when I first got Samira in Venezuela, and I started a colony and had it for like 4 generations towards the end of my career here in Minnesota. And Bill Miller helped me figure out kind of a good system in one of the growth chambers. But anyway, what you're seeing there is them feeding. It's already feeding on its yolk. Okay, so what you just saw there, and I'll repeat it, you're seeing the tracheal tubes, basically the way that caterpillars breathe, form. Let's see, let's go back to the beginning. See, I don't know about Iron Man. It's kind of like electrical. And so those little pulsings that you're seeing, that's the feeding that's the digestion occurring of the yolk. And they don't actually crap out the yolk at all. It stays in the caterpillar the whole time. And the dark spots on the head, so these are the mandibles, and these are the eyes, the stomata. Thanks. Yeah, I'm lucky to have a really good, it's not working so well right now, but I have a good system at work. Okay, so this was really unexpected. So does anybody remember or know Fred Stair? Yeah. So a University of Minnesota alumnus and future, got to be a good friend over the years because of our common love of caterpillars. And Herb actually wrote me about, there was some article and Herb was kind of like, well, why didn't they mention you and Fred Stair in this article? He's so funny. But anyway, Fred coined this term, frass flipper. So in some limacodids and in Delcerines, a subfamily, not real common, there are these little spines on the rear end that Fred thought, and they still might, but it's never really been observed, that when the caterpillars poop, these little things are supposed to flick the frass. Well, it turns out they may actually have another function in which they act like little chopsticks that help guide the yolk into the mouth of the caterpillar. So it's completely reverse of what one would think. Of course, it needs more investigation, but it's, again, part of the beauty of doing videos and time lapses that you can restudy them. You know, sometimes you might see it differently later, and that using it along with electron microscopy and other methods, it could be really good. They did an exhibit about this research I do at the Smithsonian, I guess it was about, I don't know, 7 or 8 years after I left, as an example of using research or using videos or audio for research in novel ways. So here's one that's about to hatch. And you can see this one. This is more in real time. It's chewing a little hole, so that's kind of the early stage. And notice there's no yolk anymore. And here we have a highly sped up, because it's very boring to watch it in real time, or even real time lapse. This is a monkey slug hatching. So they have kind of a problem. They basically have to get their spines to come out after, kind of as they're going out, because they're in a very thin and compressed space. So the tubercles, as I like to call them, they're not fully extended till later. So the cocoons are amazing too. So that one in number 411, that's actually the cocoon from the eucalyptus feeding one that I found at the Smithsonian years before we actually found the female. Then the bottom corner is the monkey slug, bottom right corner. Okay, so here's one of my favorite time lapses. So those little devil horns are on the back of this gelatine, another Samira. And what's so interesting, so that white that you see at the end, what that is, they spray a calcium oxalate into the cocoon matrix, and that's what makes them hard and what protects the prepupa inside. And they often don't actually pupate until months later, except for the tropical ones tend to be quicker. Here's another one. You can see where the white appears. That's the calcium spray. So it's liquidy, and it comes from the Malpighian tubules. So this one, anybody here remember Shel Sandved? Yes, Val does. So Shel actually gave a lecture at the Bell Museum in the auditorium back in the 1980s, before I knew him. Well, when I came to the Smithsonian, I heard two things. Well, I mentioned Dyer. I'd hear about his tunnels and his two families and how the tunnels connected his two families. But I'd also hear oh, you work on those caterpillars. Well, Shel Sandvid has this movie, you should talk to him. So kind of a hard guy to pin down, but I had taken film courses and stuff. So I kind of, as a postdoc, became an unofficial intern in his little office. So I used to joke about this time lapse where you can really see this is kind of the best example of seeing the calcium, even though this is from converted 16 millimeter film, So this was taken over hours, this time-lapse movie. But some of these are hilarious because you see this dark stuff going on in the background, and that's Shel Sandvid because he could never sit still. So on time-lapse, he's like moving around behind he's also the guy that does the butterfly alphabet calendar. You've probably seen that. So he did one for my son. So the adults are cool because just this general posture, different colors, the legs are hairy, they're held out in front of the body, kind of unusual, they capture your attention. And as I've hinted, the work on the development of the caterpillars and phylogeny has really been a major part of my interest. So part of it has to do with the fact, again, that all the ones that are the spiny type nettles, they share a common ancestor that fasts when it first hatches. It does not eat. Then the next day it sheds its skin, becomes a much spinier caterpillar, and then feeds. So here is a time lapse of that. And this is one actually that was not known, this species from Southern California. And if you look, see how at the beginning you have the dark spines. They're all pressed together under the skin. So then when it crawls out of its skin, they all splay out. And then it starts eating a little bit of its egg. So that's the second instar. That's first instar fasting. Doesn't eat until the second instar. They all do that without exception. And actually with iNaturalist, it's really helpful. Once you get people interested in raising them, you say, well, don't worry, it's not going to feed for a day. You haven't done anything wrong. It's going to fast. Then they know which ones will feed and which ones will fast. So here's also water a droplet of water can induce them to molt. Oh, that didn't happen. I guess it's not like, oh, there we go. So you saw it drank the water. Now it's going to crawl out of its skin. And again, highly sped up. Okay, so here's an example of a gelatine, and they start right away feeding.
Atosia himalayana.
So this is Atosia himalayana.
A lot of losses, but this one looks like it's...
And those black things on the end are frass, so it's poop. So that proves it's eating. So they eat right out of the egg. The other thing that's cool about the gelatines is oftentimes they're not smooth when they first hatch. They're spiny, but then they become smooth. So here's a great example of that. So what's amazing is these little tubercles that they have, they'll be gone after the first instar, but they feed right away. But the kind of waving around from the pressure of the hemolymph in the body is really kind of something. And this, again, has never been observed before. This is a species from Vietnam. Nobody ever knew the first stages of this one until a few weeks ago. So when I worked with Susan Weller, who eventually worked with her and Jen Zaspel on doing a smaller 3-gene molecular study, it showed what I had suspected all along, that the hag moth was actually belong with the gelatines, even though it's got these hairy protuberances that look like spines. They're feeders. So they're like gelatines that just keep those long tubercles throughout their lives, even though they can be pulled off at any time. So kind of like I showed you the lizard's tail, the pulling off of the tubercles. But it was interesting. Dyer, he was aware that they fed, but he still couldn't bring himself to put them in with the gelatines. So my latest work verifies what I found with Susan and Jen, that our hag moth or monkey slug belongs in this group with the gelatines. Okay, so now, I don't know if you know this building, Hodson Hall. So the journey to knowing lima-coated moths really began in the library at Hodson. And it was the genesis for an article that I wrote with Pam Henson, the Smithsonian historian, about Dyer back in 1990. And then the book that I'll be signing later, Moths, Myths, and Mosquitoes. So I knew about Dyer because Dyer's law of geometric growth, that you tell kind of what stage the caterpillar is by measuring the head capsule and calculating the difference. You come up with a ratio and you can back calculate, and it predicts well, in some cases, not all, but it's still widely used. And here's what Dyer looked like as a young man in 1893 when he was at Columbia. So in Hodson, I kind of discovered Dyer from the lima-coated point of view when I found all these articles in the New York Entomological Society. And so what's wrong with this picture, entomologist? On the right there, what's wrong with that? What is that scarab doing that it doesn't normally do? It should be pushing that, it's the wrong end. The scarab should be turned around the other way, pushing the ball of dung that it's laid its eggs and its larvae live inside of. They finally changed it in New York, but it took them a while. So Dyer's passion, though, for lima-coated caterpillars was apparent from when I started looking through these, and I started to find hand-colored ones, and they were hand-colored by his sister and by Emily Morton in the early ones. And then his rearing notes were quite extensive. And then he did his phylogeny, essentially a modern cladogram of lima-coateds based on the caterpillars. So even though Dyer was a major worker on mosquitoes and sawflies, it just kept hitting me in the head that there's something different about the lima-coated moths, that they were his pet group, but then I'd say, nah, he worked on this, he worked on that. So, but there was this personal touch in his rearings about putting the cocoons in flower pots and raising the caterpillars in jelly tumblers. It was very personal, and I could just visualize it. And I thought, this is just different. So basically, I've recreated the lima-coated in the jelly tumbler here. But he also described this amazing thing that you've been saying, that locomotion of just, so what it is it's muscles. They're densely muscled, and they're squeezing the hemolymph from one end to the other. So it's an open system, like all insects. So it's like a hydraulic. And that's part of why I don't like the name slug caterpillars. It's much different from the way slugs move. So I'd soon realize that knowing his fondness for these moths was the key to really unraveling various mysteries. So Dyer began his work growing up in Massachusetts and New York, but he ended up at the museum around the turn of the last century. And he brought with him his family, Zella, and children. This is a recently found picture in the Library of Congress, which I wish I had had for the book. It's tremendous. It shows actually a happier time than the one that I have in the book. So this was the old USNM building, the U.S. National Museum, where Dyer first started working. It's now called the Arts and Industries Building. And inside, this is what the crew of entomologists look like, including Dyer and various others, Caudell and Schwartz and others. And this is a much later picture in 1925 out on the mall. So as I said before, when I mentioned Dyer to Smithsonian folks, they'd always talk about his strange life and the fact that he dug tunnels. On my postdoc, this finally led me to the Smithsonian historian, Pam Henson. She was the keeper of all knowledge about the tunnels. So I asked her the name of Dyer's other wife, because he had more than one, concurrently. When she said Waleska, I realized that I knew that name from a Lima coded that he named Parasa Waleska in 1900. So that 1900 date pinned down when Dyer first at least knew Waleska. A couple years after that, I guess he created what's called a homonym. So he had to change the name of another moth to make Waleska. So it was from there that I realized, because he said it was named for Waleska Pollock of wherever. So then I went to the city directories and found her, and found that she was part of a family of teachers, kindergarten teachers, and the sister of the founder of Shenandoah National Park. So the Dyers went to Stony Man, Virginia in the summer of 1900, because that's where you'd go to have, it was sort of like a Chautauqua. It was entertaining. It was wealthy people went there to cool off from the Washington summers. So Stony Man is now is part of Shenandoah. So this was George Freeman Pollock's resort, and his sister Waleska Pollock were there in 1894 in this picture, but also when the Dyers went there in 1900. So George Pollock did start this. It wasn't officially called Shenandoah at the time, but that's basically it grew out of his resort. So later I asked Pam, well, what about Dyer's first wife? And she said Zella, and I realized, oh, that's another Lima-coated Zellans, but it was weird because it was in 1926. So you got Waleska 1900, Zella 1926, and kind of what goes here. So yeah, well after the divorce. So that, I think this was the kind of stuff that started to finally get me to want to figure out what was going on with all this. And much later, I got this picture of the Dyers in the collection at the Natural History Museum in London. Apparently, one of the mosquito workers there had visited Dyer and his first wife and requested a picture because he didn't have one, so they obliged. I guess by that time, around the time he named Zellans, they were on better terms, so this picture ended up in London. So I'm not going to talk a lot about the affair with Waleska, but they were on the same collecting trip to Colorado with Caudell, his very funny assistant, and that's Caudell's wife, Poodle, right there, who didn't think much of Dyer. And eventually this leads to, well, Dyer marries Waleska under a fictitious name, and Waleska knows she's in on it. Zella, they keep telling Zella that this other person is real, not to worry, but eventually she sues for divorce. So it's very complicated. Zella moves with the family out to California and to Berkeley, and Dyer and Waleska go out to Oakland and then to Reno to try to get a divorce. But the problem is that in Nevada, they wouldn't grant a divorce of Waleska to the fictitious person, Wilfred Allen, because they were onto it. So this made all the papers all over the country, and it was a big scandal. Okay, anyway, that's all in the book if you want to read it. Actually, Herb, that was the part Herb did not like to read very much. It was kind of hard for him. Okay, so Now, a little bit about what I learned from Dyer's notebooks. So that tiny little blue one is about this big, and there's all kinds of clues about Dyer's family in these notebooks. Pam and I had learned early on that there was a medium in his family because it was mentioned in one of these, they called them cyclopedias of biography. So when we interviewed his last surviving son from the second family, he didn't know anything about it. But when we interviewed a grandchild, one of Waleska's grandchildren and one of Zella's grandchildren, Zella's granddaughter knew something, and I'll get to that in a minute. So anyway, it's 1882, and we got Dyer referring to his limacodids as a 16-year-old, and he calls them codes or codies. And then in my book, I show some of the caterpillars that Dyer reared. So I guess pretty late in the project, when I went to the Library of Congress and looked at Dyer's genealogy notes, I realized that his genealogies that he did for limacodids in that 1899 paper were just the same style as his own personal genealogy. So yeah, in red there, I got Harrison G. Dyer Sr., who was born in 1805, who was an inventor of a telegraph prior to Morse. So that's a whole other story. But anyway, you can just see this connection. And then when I came to California, there was a family that had visited me at the Smithsonian wanting to know about Dyer. And at first I thought, oh, they're related. But then I came to realize they were, through marriage, knew Dyer's son, Otis. And so they wanted to know Dyer stuff. And as they were leaving, they said, we have these pictures of Ella, who I thought might be Zella. Well, it turned out it wasn't, but it wasn't for another 10 plus years till I finally saw the pictures when I moved to California, that in fact, he did have pictures of Zella, and he had pictures of Dyer with his son Otis with this curious sandcastle that is made like a limacodid. So in the early 2000s, I talked to my PhD advisor, Bill Miller, about all these things I was finding. And once I got to California even, because part of the evidence was out there too. And he kept saying I should write a book, as many did. And of course, they've always said you should do a movie because the story is so interesting. But then I told Bill that I had discovered this whole question about the seances and the mediums. So when we interviewed the granddaughter, yeah, she said that there was somebody at the Lincoln seances. And I thought, I wonder who that could be. So I always knew about, Aunt Parney was mentioned, his mother was mentioned in his notes. So there's this Aunt Parney that collected tiger moths for him back in 1889. And then his mother, I later found out, was a trance medium. But what was interesting is I knew that Dyer's mother's maiden name was Hannum, so I put Parney, Hannum, Lincoln, seances as a Google search. Wham! That's how I found him, was from the notebooks to Google. And then I was able to corroborate it by going to Library of Congress, finding all these letters from Aunt Parney, all this stuff about his mother in Boston as a trance medium. So it was a family of spiritualists, and really interesting. And I could write a whole book just on that. So now a little bit about the tunnels and his battles with entomologists. So this is a Minnesota thing. You know, I guess at one time the Washington team was called the Griffman or the Griffiths, after the Griffin family, who you know, Griffith family, who were the owners of the Minnesota Twins eventually, that had moved from Washington to Minnesota. But anyway, Walter Johnson had 13-game winning streak, and So this was kind of a slow time in Washington in 1924. So at the same time, or September, excuse me, so a truck in an alley caused a collapse, and it turned out there was a tunnel underneath there. And there was all kinds of, it was a labyrinth that went for blocks, brick lined and all. So it made it into the newspapers. Headlines, old tunnel here believed to have been used by Teuton war spies and bootleggers, so all kinds of stories. There was a political satire about fighting Bob La Follette in Wisconsin, who wanted to enter the White House through the tunnels. I actually was at Rocky Mountain Biological Lab with Doug La Follette, his grandnephew or whatever, who is a conservationist and former Attorney General of Wisconsin. So the next day, the mystery led to Dyer. So he was interviewed in the newspapers, and he said it was a pastime, not anything nefarious, just a hobby. Like he had enough time to do all that. And yeah, no computers. Oops. So yeah, he was, let's see. I know sometimes these videos work and sometimes they don't. There we go. Yeah, oh well. Yeah, here he's showing how he laid the bricks and stuff. I got a couple different pictures from the same, a different interview, but same. So I was able to animate the pictures of him. But anyway, this is what his second set of tunnels were. So those ones collapsed in DuPont Circle, but then he was very proud of his current set of tunnels, which were 3 stories underground. So this shows you kind of in relation to the house, which was across from the Smithsonian, where the tunnels were on the side of the house and down. And fortunately, that was the beauty of the interview with the last surviving son, is he actually helped his dad with the tunnels. So he had firsthand knowledge of hauling the dirt out. And then the grandchildren had heard stories, but they were secondhand. So this shows where the 2 sets of tunnels, and the story about Dyer is he had 2 families, and he went back and forth between them by these tunnels. And of course, that's apocryphal, and they're different times. They were done, or different places, different times. What's really interesting, though, is that the grandchildren, as part of the fact that there was some contact between the 2 families, realized they each had a child that didn't fit with anybody else. So they actually did go to school together, much like the Dyer kids were supposed to have met each other, that they each had a dad that worked at the Smithsonian, so that's how it was figured out that the 2, no, but one generation removed, it's like the myth had legs, and it's like it actually happened. So when I interviewed, these 2, it was really quite something. So the other big story about Dyer is with Smith and about their competition on moths and mosquitoes. So the legend was that Dyer named a species corpulentus or Smithiformis after Smith, who was heavy, and Smith was to have countered with the name diarrhea for Dyer. And Dyer's son had never heard this, of course, and he just thought that, he just wanted me to keep telling that story over and over. But it actually didn't occur that way. The true story is a guy named Berthold Nemoigan, who was a friend of Dyer's. He was a Jewish banker. I've done biographical research on him too. Had an amazing collection, but he named diarrhea, he didn't think of it as diarrhea, as a genus after his faithful friend, or at the time, Mr. H.G. Dyer. So, let's see, yeah. So in Ent News, Skinner, who also became kind of a combatant with Dyer, he poked fun at the fight between Dyer and Smith after a trip to Dublin, New Hampshire, and actually wrote a poem about it, which is really humorous. but each one claimed to be the discoverer of the mosquito breeding site. That's Dyer panning for mosquito larvae, by the way. That's a movie that I created because I had stills. So, in fact, it wasn't Dyer or Smith that found the breeding sites. It was their able assistants. So that's so typical of people taking credit for what they didn't discover. So Dyer wrote to Skinner at some point in one of his battles, "I'm rather surprised you should have allowed yourself such a hysterical outburst. Better take a sedative." So these kind of went back and forth. Dyer was incredibly productive, though, and so one of the challenges of writing this biography was to kind of give context of his contribution, but by the same token, kind of tell the real story, and of course to document it, which took about a third or fourth of the book, just all the, going to Library of Congress, to the National Archives, Smithsonian archives all over the country and the world, really. So it's a compilation of so much research. And I started it when I wrote the article in 1990, but since I was employed after my postdoc by the Smithsonian, I continued piecemeal over the years and pulled this all together. So this quote is apropos. "Ah, well, great oaks from little acorns grow." And of course, that was true of his tunnels, was true of his contributions. So here's a little music. A little background on that. So this is a song I wrote called "Glide on the Leaf," and it's actually about the locomotion of limacodids, of the caterpillars. And I came up with the song idea after I got rear-ended by a car. Thank you all.